All right, so the DNA is more, oh, you're all still there, right?
Yes.
Yes.
The DNA is more complicated than we thought, or you know, so here's the promoter, right? Like I was talking about, and then the DNA all gets copied, okay? The template strand, and so when it's transcribed, we call that full length of RNA that gets made the pre-mRNA because it hasn't been processed, and so the promoter isn't copied and the termination sequence isn't copied, but everything in between is. And now the RNA has to get processed, and it's processed based on which proteins the cell has signaled need to be produced. So a single gene can code for more than one protein based on the processing of the exons and introns. So there's a couple of things that happen here. There's a protein cap and tail that get put on, and the tail protects the RNA from getting like chopped up and losing part of its sequence for the protein on the way to its destination. And the cap is basically a zip code that says where in the cell that RNA is supposed to go. Because we think about, oh, the cells are tiny, how could this thing possibly get lost? But it's going to be sent on like a factory conveyor belt system through the cytoskeleton. If you can kind of picture the cell as actually a really humongous factory, and which, you know, UPS truck or Amazon truck is that RNA supposed to end up at, and is it headed to Florida or Maine or Washington or, you know, Mexico, right? Where is it going to go on its journey? And that's going to be determined by the zip code in the cap, which sends it to the right place with the cytoskeleton. The other thing that happens is, you see how we have introns cut out? And I really don't know why they're called introns instead of exons. It seems kind of backwards to me, but you can maybe remember that based on the humor. So certain introns are removed and the exons stay in, and the RNA gets spliced together based on all the exons and whatever introns didn't get cut out. And so now the sequence of that RNA has changed from the original DNA. And so the codons aren't exactly the same as the original DNA, but it's still going to determine what polypeptide gets made. And so the finished, oh, that wasn't what I meant to have happen.
How do you know if it's an exon or an intron?
They just gave them those names based on what gets cut out and what stays in. I'm sure you can imagine that there's probably a couple-hour conversation in detail we could have about this process. But the things that I want you to know is that transcription occurs where we make an expendable copy of the DNA, we make the RNA, and then the RNA gets processed, and as processing, we get a cap and a tail for protection and for a destination. The cap is the zip code, and we also splice out certain segments of the RNA in order to make the final finished mRNA that's going to code for the protein. I don't know why it's introns versus exons. Somebody was having fun when they did that. Maybe they were drunk. I really don't know.
Okay. But we don't need to know which like pairs need to come out.
No. This is just generic. You need to know the RNA has to be processed so that it has a destination. Okay. And you know, this slide just gives you in a nutshell why I care about the cap and tail, and how is it possible for a length of DNA to code for more than one protein. It's based on what introns are cut out and which ones are left in with the exons. That's all. Okay. So that is transcription, DNA to RNA. All right. And translation is how we use then the RNA molecules, the 3 different kinds, and the proteins that combine with our RNA to make ribosomes, to then translate or read that mRNA and synthesize proteins. And it has 3 steps: initiation, elongation, and termination. And this is where, here's the reminder that these are the 3 types of RNA that are required. So that's a repeat slide. And then where does translation occur? Wherever there's a ribosome. So there are ribosomes in mitochondria. There are ribosomes on the surface of the rough ER. There are ribosomes in the cytoplasm. Wherever ribosomes are present, there are ribosomes on the surface of the nuclear envelope. Wherever a ribosome is located is where mRNA and tRNA can combine together with the ribosome subunits to translate mRNA into a polypeptide. So this slide is a reminder that ribosomes have two pieces, the small and the large, and that it's made of rRNA and protein. And they have to join together during translation, and they have special binding sites for both mRNA and tRNA. So this is where we see all the RNA types together in translation. This is kind of an interesting model that shows you trying to make a 3D shape to show you the small and the large subunits joined together, and where the tRNA molecules can come fit inside the ribosomal complex, and then how the mRNA can be fed through the specific like gap in the subunits, and then as those tRNA molecules come in and out, there's a polypeptide chain that grows together. Okay, and this diagram just shows it more as a 2D that shows the small subunit and the large subunit and how they join together, kind of like a hamburger bun, right? And in doing so, they create these two spots for tRNA to temporarily join up. And then I'm gonna skip, this one actually shows you, hey, what does tRNA look like? And it's a long chain of RNA, so it's a nucleic acid, and it folds on itself, and it has hydrogen bonds between the nitrogenous bases. But at one end, it has a 3-base sequence that is free, that can attach to the amino acid. And then in the middle of this loopy thing, it has a 3-base sequence called the anticodon. And that anticodon is going to be where the tRNA molecule can match up and temporarily bind with the messenger RNA. Okay, so it has two sites for binding, the amino acid site on the end and the anticodon here in the middle. And if I just kind of smush all of that together, it sort of looks like a firecracker, right? You've got this end that's the amino acid attachment site, and this end that's the anticodon binding site. And so how is it then going to be used? So you'll see a lot of different diagrams that kind of show you a tRNA molecule. But in this particular one, it says, okay, here's the large subunit, here's the small subunit, and as the mRNA gets pulled through the ribosomal complex, the tRNA molecules drop into the binding sites and they bring their amino acids, and the polypeptide chain grows. So the writing on the slides just walks you through what I'm going to do, kind of remind you of. So here's the first row. So on the, and steps 1, 2, and 3 in that first row are all what we call initiation. It's a sequence that happens in order that gets the complex all ready for the polypeptide to be processed. Here we are back in the game. So basically initiation is a 3-step process where the first thing that happens is that tRNA binds to the small subunit. And we call that the pre-initiation complex. And the good news for you is that you are going to be able to look at the diagram when you take the test, and you don't have the you don't have to memorize this and label it for memory like I make my students do in person. So there's one perk of doing this online. There's no point to making you memorize it and label the blank diagram. I know I'm mean, huh? Okay, that'll be on YouTube forever. Okay, so then the second thing that happens is because that first tRNA that always has methionine, it now has created a spot for the mRNA to come in and bind. And so the AUG, the start codon, comes in and matches up with the anticodon that has methionine, and then step 2 is done. And the third and final thing in initiation is that the large ribosomal subunit can now bind to the small subunit. Now there's some details behind the scenes and how that biochemically is made possible, but for the purposes of 160, it doesn't matter. You just need to know the sequence. That initiation is just all those pieces coming together in the right order that now have created tRNA binding sites. And there's a protein site, because if it's a catalytic site, you can bet an enzyme is involved. There's a catalytic enzyme site in the large subunit that is basically going to create a chemical reaction that transfers the amino acids from one tRNA to another. And so that the tRNA molecule that entered first transfers its amino acid over, and then it leaves. And that's what you see in elongation. Okay, so there's a couple more slides that review verbally, you know, the process for you about what happens. So steps 4, 5, 6, 7, and 8 are all elongation. And step 9 is termination. So this slide looks at 4, 5, and 6. And basically, elongation is quite simply about, for however long the mRNA molecule is, tRNA molecules will come into those 2 binding sites and drop off their amino acids, okay? So essentially what happens is that first tRNA already has that first tRNA binding site full. And then whatever the next codon is determines which tRNA molecule with its anticodon that's complementary can come drop into the second tRNA binding site. And the catalytic site catalyzes the breaking of the bond between methionine and its tRNA and the reformation of a peptide bond between the first and the second amino acids. And as soon as that happens, this tRNA molecule can no longer bind anticodon to codon, and it gets kicked out, and the ribosome ratchets down one step. And so now the tRNA that was in the second site is now in the first, and the second site is open. So can you picture that happening? Repeat, 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 repeat every time. See how now we have a new tRNA molecule that comes in and its anticodon is complementary, and that dictates which amino acid is going to come in next. And the catalytic site catalyzes the transfer of the amino acids over to the one that just came in. And so we have this growing polypeptide chain where it always gets transferred to bind to the most recently brought in amino acid, and the tRNA that's in that first binding site gets kicked out and it ratchets down. So we could have 1,000 of these, right? This could happen 500 times, 1,000 times, however long that polypeptide chain is, however long that mRNA is, it slides down until we get to a stop codon. And what happens when we reach the stop codon is there's no tRNA that can bind to the stop codon. The stop codon actually signals the release of that ribosomal complex, and the large and the small subunits separate from the mRNA, and the completed polypeptide is not bound to anything, and it now needs to then go get processed into a functional protein. So just to recap, what is initiation? It's all of those parts coming together in a particular order to create the tRNA binding sites. What is elongation? It's the pairing of the anticodon of the tRNA with the complementary codons of the mRNA to bring the amino acids in the sequence dictated by the messenger RNA. And that moving through the first and the second tRNA binding sites as the catalytic site catalyzes the elongation of the polypeptide chain. And then what happens at the end? We reach the stop codon and everything disassembles. And guess what all those parts are ready to do again? They're not used up. So the mRNA could go right back around and join up with another start tRNA that has the methionine bonded to it and the large and the small subunits, and do this 1,000 or 1,000,000 times over until there's some cell signal that says, hey, that's enough of that particular protein. And then all of that stuff gets disassembled and well, really what gets disassembled is the mRNA, and the ribosomes move on and do the job of translating the next mRNA molecule. So this just recaps transcription and translation for you. This one is showing you how we, where transcription happens, that it's making the RNA, and then it shows you how the amino acid is going to attach to the tRNA molecule, and then it's available to be a part of translation. So then 3 shows you initiation, and 4 and 5 just walk you back through elongation and termination. So it's just a different visualization. There are probably 100 different models out there, okay? So that is all that rest of that PowerPoint is about, is transcription and translation. So if you watch a couple of videos, or you have already, and you do the homework, there's nothing else that I'm trying to throw in there. It's just, do you understand the steps of the process: initiation, elongation, and termination? Can you match transcription and translation processes to the actual thing that's happening, as opposed to accidentally swapping them, right? And then with the lab, you actually get practice with the base sequences and what that looks like. So here, looking at this unlabeled diagram, this one here in the upper left-hand corner, if I make this big, thank you. My 5-year-old is ratting out her brother. I don't frown upon that because he has little self-control. Okay, so what I wanted to show you here was look at 1, 2, 3, and 4. Can you identify what those structures are?
Red cycle number 2?
No. We went through this one, but in this picture right here, see how this is just a blob showing as a ribosome? Do you know what is going through here? So it's just a different schematic. So here, here's the ribosome. 4 would be a codon, something that codes for an amino acid. And see this spirally thing? This is a tRNA. And here's its anticodon. And then what is it bringing in?
Sorry, where's the anticodon?
The anticodon is these right here, UGU, right? Okay. And then over here, you see these guys, this one like should get kicked off by now, right? But these guys right here, what did they just bring in? This one just brought in arginine. This one has phenylalanine.
Amino acids, right?
Yeah, and then what is this 2? What's growing here?
Polypeptide.
The polypeptide chain. Yeah, so it's just like, can you take what you know and look at a diagram and process it? And so that was just me.
What was number 1?
An amino acid.
Okay.
Okay, so that we've already walked through this one, and I'm going to close this. And 16 is meiosis, and 17, if it will expand for me. just going to change it to one page and zoom in. Ooh, that got really big. Okay, so does this first diagram looks familiar, right? Reminds you where transcription and translation are occurring. And then based on what you've read and what you get reminded of in the document, I think you can come up with differences between dRNA and RNA, like which one is double-stranded, which one is single-stranded, which one uses uracil, which one does not, things like that, okay? Then figure 2, just as you read the figure and remember a little bit about the processes and correctly identify transcription and translation. So DNA to RNA, transcription, RNA to protein, translation. So if you remember the base pairing rules, do you remember what the U in RNA pairs with in DNA? Well, what do you see it across from?
A.
A, okay. So there we go, first page done. So then, just a little bit of review here. How many letters of RNA are needed to code for one amino acid?
3.
3, and do you remember what that 3 base sequence is called?
Codon.
A codon, good. Okay, what does transcription make?
Trouble.
It's the
RNA.
first thing. It makes RNA, good. Where does this occur?
Nucleus.
Yay, all right.
We're getting
Right.
it.
So if we're gonna transcribe this template, what pairs with A? What's opposite?
For the RNA,
For RNA,
a U.
U, and opposite T would be
A.
A. Opposite C is
B.
B.
G. Opposite G is
C.
C, and then opposite A would be
U.
U. Then opposite T,
A.
A. Opposite A would be U, and opposite C would be
G.
G. So if the sequence were read from left to right, this would have to be like the, well, that would make this like the promoter sequence, right? Or something that was going to get, you know, we don't start anything because it's not until we get to what?
AUG.
The AUG that it's actually part of the mRNA, okay? But then we've still got to, you know, we're still practicing transcribing it, and you've got to recognize that that was a promoter sequence, but it's not a big deal. I didn't ask you for that right now. I'm just pointing out that, hey, you know that where's the mRNA really going to start? It's here with the AUG, okay? So this particular diagram is just showing you, hey, inside the nucleus, what are we doing then? If we're inside the nucleus, transcription or translation?
Transcription.
Transcription, yep, and it walks you through, and it's talking about the transcription initiation complex because of the RNA polymerase binding with the DNA template strand, and that there are also transcription factor proteins that help this process work, and it basically moves the polymerase can then read along the open section of DNA and make the RNA. And so it just shows you that there are free nucleotide bases in the nucleus that get grabbed by the RNA polymerase and get used to make the strand of pre-RNA. So that's all happening in the nucleus. So you have to remember, you know, walk back through this and read it and answer the questions about it, okay? I made it a little smaller so I can scroll without getting dizzy. Let's see about this one here though. In which direction is the pre-mRNA molecule constructed? This has to do with the 3', 5' business.
5 to 3.
5 to 3, yes. So we
5
always,
to-
we're synthesizing 5 to 3. DNA gets synthesized 5 to 3. RNA gets synthesized 5 to 3. We read
That's on the new strand, right? So on the parent strand, it's 3 to 5.
it. So DNA gets read 3 to 5. But DNA and RNA get synthesized 5 to 3.
Okay.
And then here's something a little bit tricky. When tRNA brings the amino acids, the mRNA is getting read by the ribosomes 5 to 3. So just think whenever something is being synthesized, we're doing 5 to 3. DNA and RNA get synthesized 5 to 3. And when the polypeptide is getting made, the mRNA is going through in the 5 to 3 direction.
So it's being kind of read by the ribosome?
So, well, the ribosomal complex with the tRNA is essentially reading it, yeah.
Okay. Unzipping, reading,
You know, all of that
same
business.
thing.
Okay, so looking at this right here, just something for you to think about. Considering that many types of cells in a multicellular organism and their different functions, knowing that every single cell has all of the DNA and all of the genes. Are all cells going to read all their genes, or are some of them never going to be read?
They won't all be.
Yeah, because there are certain proteins you never need in your liver cell, but you do need in, your heart cells, right? So while the genome is the full complement of a cell's DNA and an organism's DNA, and the genome is contained within a single cell, the whole genome is not expressed because of differentiation, as in specific expression of certain genes that make a particular cell, its cell type and its tissue type. Otherwise, all the cells would be the same. Okay. Figure 3 has you think a little bit more about translation and where it's occurring. And then this next one reminds you about the mRNA, tRNA, and rRNA, and asks you to look at figure 4 and what it shows. So you just have to, explain that you see that. Look at the DNA strand here. Some of the genes code for what? Making tRNA. Some of the genes code for
mRNA.
making proteins. Some of them for making rRNA. Some of them for making messenger RNA and the way the RNA is going to be used. Okay, so if you look at this, yeah, you're getting revealed another type of RNA.
I know,
Even
it's
though
like-
insisted, like shh.
Typo.
No, it's not. It's just way above your pay grade. So
Learning to embrace that.
when you probably previously thought about DNA, you probably thought, oh, DNA codes for protein. But now you see that some lengths of DNA code for individual lengths of RNA. And some of the RNA is tRNA, some of it's mRNA, and some of it's rRNA, and some of it's another kind of RNA you don't even want to think about. And some of the DNA strands don't code for protein at all. They are promoter sequences and termination sequences. But they're all part of, so could you imagine that this particular length of DNA has all the codes necessary for the tRNA, the mRNA, and the rRNA for a particular protein or set of proteins to be made? That we don't keep the tRNA for one protein on another length of DNA, but that they're all going to be, close together. That's just something to make you think about. But DNA is really complex, right? And My daughter found something interesting on her tablet. Sorry, I was just hitting delete. So I just want you to think about how DNA is a place where genetic information is stored, and it codes for all of the molecules needed to make those proteins, not just the protein itself, okay? And then the next one walks you a little bit back through RNA processing, like we already went through, so that should be a breeze for you, having gone through this. But just think about this one though, because we talked about it. How can a human genome contain 25,000 genes and yet make 100,000 different polypeptides? What method did I give you for us to get 4 times as many proteins as there are individual genes?
Where are you?
Page
30.
I don't even know. We're on number 30, but I'm not sure what page this is.
Okay.
Like page 3 or 4. The diagram, well, it says model 2, but
Okay.
what are you seeing here in the RNA? Introns and
It's a different
Exons.
combination.
yeah, so different spliced combinations of the introns and exons will lead to a different RNA sequence that can code for a completely different protein. So on average, a gene must code for something like 4 different polypeptides, some more, some less, just to get, you know, the 4 average.
The average gene codes for 4.
For 4 polypeptides,
Wow.
yeah, because 25,000 times 4 is 100,000. I already told you about this tail and what it was for. All right, so that is, here's the codon table. And you just have to kind of walk through it a little bit. And remember, on the test, you need to just know how to use a codon table. You don't have to have it memorized or anything like that. Okay.
How do you use it?
How do you use it? I
Yeah.
think maybe what, okay. So here, we'll do 38. And then I think what I need to do is make a little mini video to do part of the lab, because we always do the lab, this particular lab, we always end up doing it together. But if you look at 38, what I would say is, okay, opposite T is A, opposite A is U, opposite C is G, and then opposite C is G, opposite T is A, here's another A, then I'm going to have GCC opposite the CGG, and then A would be U, T would be A, and the G would be C, and then GTC becomes CAG, and ACT, the last one is UGA. So now what I do with that, and I'm going to shrink this down a little bit, is I have to okay, it's kind of not, it's far down here. So I'm going to copy my strand just down here so that you can see it, okay? And now, what do I know as far as the amino acids that it codes for? So what is my polypeptide from that? So this is, what's the polypeptide sequence? So I find AUG, and here's A, here's U, and the third one, AUG, is methionine, the start codon. So I would write MET. And then I find GAA. So the first base you find on the left side. So here's G. Then I go to the column that has A, and then within there I find the third one. GAA is, GLU is the abbreviation. That's glutamine. It doesn't matter that you know that. I just happen to. Then the third one, GCC. So here's G. Second base is column is C. And GCC, the codes for ALA, which is alanine.
Okay.
And then UAC, here's U. Third column is A. And UAC is for TYR. That happens to be tyrosine. Then CAG is number 5. So here's C, here's A. CAG is for GLN. I think that's glutamine. And then UGA, here's U, here's G, O, and A. What is that?
Stop.
Stop. Okay. So we have a very, very short polypeptide sequence that we have just decoded from the mRNA using the codon table. So that's
Okay,
how you would
thank you.
use it. The lab has you practice and make sure you do the 5' and 3' in there. And what I think might be helpful is if I just made a short video where I just take one section and walk through a little bit of it, because you'll be turning that stuff in and then you have to turn around and take the test.
Coronavirus.
You need I'm doing a really short one this week and I need to post the picture and I've got to find a little,
Okay.
There's a single reading and it's just about how the coronavirus gets into cells. So I was trying to find the right short article that I wanted you to do, but I made sure that lab module 8 was up. Okay,
Yeah,
so
thank you.
yeah, and I still have to put in the post test for 7 and for 8. For learning assignment 16 for meiosis, that is pretty straightforward. Walking
It's
through,
pretty easy,
practicing labeling and identifying. So what I promised to do, and this is kind of at the end here, so people can pause when they're watching the video or watch me ramble on if they want. I'm opening up both of the labs. What you see on the left-hand side, all those Word documents, those are for my my other job that's very much part-time, which is just online grading. Oh, I snagged one of them on accident. There we go. Okay, so here is 7 on the left-hand side. I'm just going to make it a little bit bigger here temporarily. And once the pre-lab part is a quiz. And then the lab part, as we were talking about, this overview worksheet is just the short one. And if you do the long one for extra credit, you can put the long one in its place. So I'll make a little announcement. Instead of an announcement, I'm just going to drop it into the directions on the lab page, okay? And then, because normally we do this in class, so I not only show this labeled, but I talk you through it. Instead, I told you what they were, and then you have to match late interphase with the definition or the description that describes what happens at interphase. So this helps you see what's happening with the cells. I wouldn't try to print this and do it from it as a printed version, but it lets you know that, hey, D is anaphase, where I talked about an actual image of a cell where the sister chromatids are splitting in mitosis. And so then I would say, all right, so if D is anaphase, what do I know is happening during anaphase? And I'm going to match it up here where, oh, 3, the sister chromatids separate as the cell elongates and the kinetochore microtubules shorten. So in a nutshell, I've given you the key things that are happening during that point of mitosis or interphase. And so I would just put a 3 right here to match it up. And so you're going to do that for all of them. For 2B, there's not something you turn in. You use what you do here to do 2C, okay? And you can either use actual items or you can come down to the end here. Oh, into the appendix, and you can print the dark and the light, or you could draw your own. This is for meiosis. It's the second thing. Okay, so you can make your dark and your light copies, or you can use the bricks or the blocks, or you can draw your own. And that's why there's a couple of different options there. So scrolling back up, making people who are watching the video ill. What you're essentially trying to do is then use your items or your strips of paper to set up a scenario where you show the sequence of mitosis. And basically you're going to take a picture or draw it and put it in under 1, 2, 3, 4, 5, and 6. Okay. And so it's just you practicing drawing this out. Okay. And so you've got a lot of pictures of what that looks like. And in fact, on your learning assignment for mitosis, I believe you have a bunch of images of that to work with. Plus we went through it in detail. And it says, hey, label the chromosomes, spindle and centrosomes, and you're drawing. If you're actually going to make a mock cell, what you would want to do is have materials that you could use to show as the chromosomes, the spindle, and the centrosomes. And then step 3 is just answering a couple of questions. And then here, now you get to practice. And again, having it on your computer screen is going to be much better than if you were to try to print this. So when I look at number 1, I see that they are separating, and now I have to decide, is that anaphase or telophase? And I might decide that I'm going to make this really big on my screen, right? And do you know what I can see here? This is an onion, a plant cell, and what can I see in between? It looks like there's maybe a cell plate.
Cell plate.
A cell plate, and you notice the tips of the chromosome, the chromatids aren't so visible here.
Yeah.
So I might be more likely to call this telophase than I would anaphase. Number 2, it's a question about, what do I call that? Prophase or what? But look at 14. This one is for sure
Metaphase.
Metaphase, okay? And then look at 12. And 12 might help you decide about 1. Do you see a definite cell plate across the middle here?
Yes.
12 is telophase, and look at 16. 12 is also telophase. So I might be like, oh, 1 is maybe anaphase, maybe telophase. And 11, I would be like, well, I can see the cell plate here in 12, but I can't see it in 11. So is this just the very, very end of anaphase? And 9, do you see them separating? Maybe, or is 9 prophase where they're coming together? Do you see how there's a little bit of wiggle room here sometimes to identify what step something is in? Okay, so what does it say though? It says it might help to magnify. So I zoomed in. If you aren't sure of the phase, because these will be judgment calls, you explain your reasoning. So I would be like 6, look at 6. 6 is anaphase because I can see the cell plate. But then I might say 20, 6 is telophase because I can see the cell plate. But 20 is anaphase because even though the sister chromatids are at opposite poles, I don't see the cell plate, okay? And then for 19 here, I would say, oh, well, is that prophase or is it metaphase? Do I think they're lined up in the middle? It's really, they're kind of lumped together. So it's either prophase or metaphase, but which one is it? And then I look at 14 and I'm like, okay, I can see those chromosomes are for sure lined up in the middle. So 14 is metaphase. So just explain why you think it is what it is. And you're not going to be marked wrong. You just need to explain yourself, and it gets you to think about what are the things that help you identify which part of mitosis the cell is in, okay? And then the last thing that you do is meiosis, okay? And I want you to think about how the genes in the parent cell are all diploid. But when the reproductive cells make gametes, the gametes are haploid. But as soon as fertilization occurs, what are all your cells for the rest of your life?
Diploid.
Diploid, exactly. So the only time you have haploid cells is when we're talking about the gametes that you have, okay? Only the eggs or the sperm are haploid. Everything else is diploid. And so that's the introduction. And then you get a struggle a little bit here with meiosis and what happens with the steps, and how this one shows you the first round, so columns 1 through 4 and 5, sorry, and then the last 4 are meiosis 2, so the first round and then the second round of division. And then what you get to do is practice. So this is where the last thing, you could model this with the strips in the appendix if you need it all the way at the end. If you want to print this last page and move things around, it might help you. But if you can do it visually, then you're just separating. So what would happen here? Oh, I don't know if I'm going to get to, this is not PowerPoint.
Does it go through like meiosis 1 through prophase, metaphase, anaphase, and then start all over with the second, or does it go?
Yes, it does. So
Okay.
here we would split, right? So at the end of meiosis 1, we're gonna have this chromosome over here and this chromosome over here. And then what happens at the end of meiosis 2 is that I have one sister chromatid, AS here, and I'm not sure if it will let me.
And when they're in, before they split into 4, when they're in the 2 separate ones, that's homologous chromosomes, right?
Yeah, so here, if I were to show it, this is getting kind of fun. Okay, so I've attempted here. Uh-oh. That's not what I wanted. We'll just undo
That's
that.
what I was doing on the pre-assignment, trying to move around.
Move things around. It's hard. You could always redraw it and then just paste in your own diagram. That's totally fine. So what happens here is the homologous pair separates in meiosis 1.
Yeah.
So at the end of meiosis 1, we're haploid because although we have replicated DNA, how many of the chromosomes do we have of the pair? 1. So at the end of meiosis 1, we're haploid because you only have 1 chromosome from each set. And at the end of meiosis 2, we've separated the sister chromatids. And so this would be 1 chromosome with little a, little s, 1 chromosome with little a, little s, 1 with big A, big S, 1 with big A, big S, which always cracks me up. And then, so round 1, we separate the homologous pairs. Round 2, we separate the sister chromatids. And that's not even thinking about crossing over. And so then you're going to think about crossing over and what happens if a length of the DNA swaps, then the particular gene versions could swap. And so I want you to, well, think about what happens. We know you'd have big A, big S, but what about big A, little s, and little a, big S? So that's what these become, is this one, little a, sorry, big A, little s, big A, big S, little a, big S, and those are my options with crossing over, because these could swap, these non-sister chromatids could actually swap, okay? And now you get to think about, if there's no crossing over, but we have more than 1 chromosome set, what happens if at metaphase, the orientation of the second pair is different? Yeah. So here, straight down the middle, what would we get? We would have that, right? Or
that's really small.
Yeah, it is. Let me see if I can. Oh, come on. I highlighted you. Cooperate. There we go. How about that?
Yeah.
So down here, you would have, oh, that shouldn't be big. It automatically capitalized it. We would
Oh.
have these two genes, right? This, you'd have this chromosome, and you're going to have that chromosome, or you're going to have this chromosome with that chromosome, okay? Without any crossing over, the genes stay together. And then that's here, okay? And then when they separate,
Are they lowercase L's?
oh, yeah, sorry, thanks.
It's hard to know.
So then that's at the end of round 1, and at the end of round 2, you would have in each gene, oh, it's gonna do that for me. Oh, I know, this is what you have at the end of round 2. So at the end of round 1, you basically, you've got, it's easier when I can draw it. So this would be, both sides would be little a, s, right? Little a, little s, little a, little s, and this would be L. And then when it splits in here, we would have one side of it that would be AS, little a, little s, and one that would be little L. And over here, it's going to be big AS, and then the other one is big L. And over here, the same thing. But what happens over here, so I end up with, so this is the way I should have done it, is because you basically have, you have this on the two different sides, right? If we're showing the sister chromatids together, and so then at the end you have AS, you have that option. Or, I'm getting really annoyed with my computer right now. This is what you have, and I'm drawing a line under it to show that that is an individual chromosome, okay? So here in the first row, I've basically shown that we still have the sister chromatids paired together at the end of meiosis 1, but I only have one chromosome or the other. I don't have both. And so this is gonna be a little bit too big for what I want. And over here, what I have is do you see what happened here?
Oh, because they lined up differently. They lined up differently.
Lined up, yep,
Okay.
they lined up differently. Good. So I have this. So this is here, okay, where this collection came down, and this collection came down, and then what happens at the end?
They swap spit.
Right. Well, I then have. See how they're different? Because of the swapping places at metaphase. So even without crossing over, meiosis introduces variation because of the independent way that this chromosome pair lines up at metaphase. It doesn't matter which orientation this one has. This one can do what it wants. And if you have 23 sets and 2 locations, 2 possibilities, left, right, or right, left, if you follow, then the genetic differences you can get in the gametes is 2 to the 23rd, which amounts to, here's my little sum here, more than, I'll highlight this here, more than 8 million different combinations. 2 to the 23rd is greater than 8 million, just by independent assortment. And that's why you might look like a sibling, but not exactly, even though your genes came from the same parents. So hopefully that was helpful. All right, we
Okay.
are,
Tell me about the test. What are we looking at? How many minutes? How many problems?
hold on a second. Stupid computer.